This is video number 11. We continue talking about monosaccharides, precisely the cyclization of monosaccharides that have more than 4 carbon atoms. I mean pentoses, hexoses, but also heptoses. We don't focus much on heptoses, but all monosaccharides that have 5, 6, or 7 carbon atoms will prefer to adopt a cyclic form just because it is more stable than the linear form. The process of cyclization, you are not supposed to know any detail about it. What you should know is the fact that cyclization may form a 5-member ring or a 6-member ring. Okay? Of course, the 6-member ring or the 6-member cycle can be formed only by hexoses and heptoses. However, the 5-member ring, a cycle of 5, may be formed by pentoses, but also it may be formed by certain hexoses. Fructose, for instance, prefer a 5-member ring. Let's now forget about the details of cyclization. We don't focus on the chemical reaction that it transforms this molecule, the linear and cyclic one. We must remember that when a certain monosaccharide, for instance ribose, adopts a 5-member ring, forever it will prefer this form. If glucose adopts a 6-member ring, forever it will prefer this form. We are not going to memorize the cycle the cycle type for each monosaccharide, but only for ribose and hexoses we must know that ribose forms a 5-member ring and hexose and glucose forms a 6-member ring. These chemical structures must be known because they will help us learning and memorizing other details that come later on in other chapters. Let's now name these this type. These are named furanoses. All monosaccharides that will form a cycle of 5-member ring, they will be named furanoses. 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 And those that form a 6-member ring, they are named pyranoses. So this is another classification based on the criterion of cycle shape. In addition to the cycle shape, we have another important isomer that is formed when cyclization takes place. Cyclization takes place by reaction between hydroxyl group that is carried on carbon 5 for hexoses or carbon 4 for pentoses, doesn't matter, you don't memorize these details. It is a reaction between this oxygen and the carbon atom that carries the functional group. Of course, C double bond O, it is the aldehyde group. When this oxygen reacts with this carbon, the oxygen here will become hydroxyl OH. It is in the blue box. This OH was the C double bond O. Now that the cycle is formed, there are two options. The hydroxyl group may be directed downwardly relative to the cycle, the plane of the cycle, or the hydroxyl group may be oriented upwardly. Don't ask about what is responsible for preference of downward orientation or preference of upward orientation. There are many chemical considerations. We are not supposed to explain them here. What you should know is only ribose forms a furanose. The furanose may have the OH directed downward. It is named alpha isomer. If OH is directed upward, it is beta isomer. Exactly the same with the glucose, except that we have a 6-member ring. It is a pyranose. Hydroxyl directed downward, hydroxyl directed upward. This is alpha glucose, this is beta glucose. This is alpha ribose, this is beta ribose. Now we go back to the polysaccharides. Remember, I told you that monosaccharides don't combine much. Glycogen, starch, and cellulose are all polymers of glucose. What is the difference between glucose that forms these polymers? Actually, glycogen and starch, of course you remember that glycogen is present in animals, in a human, in the liver, in the muscles. Starch is present in plants, potato tuber for instance, banana fruit, many other examples. Starch and glycogen are polymers of alpha-D-glucose, while cellulose is a polymer of beta-D-glucose. If you take this, make a polymer, you get cellulose. If you take this one, make a polymer, it is either starch or glycogen. I hope you know that we can digest starch in our stomach, which is a polymer of alpha-D-glucose. We cannot digest cellulose in our stomach, which is polymer of beta-D-glucose. So the difference is really significant and it is important to be known. Let's move now to disaccharides. Actually, we go back to the first classification, started by classifying carbohydrates into monosaccharides, disaccharides, trisaccharides, oligosaccharides, and polysaccharides. I go back to this classification. We have actually three examples of disaccharides we must know. We must not memorize the chemical structures, but we must memorize the monosaccharides that form each of these disaccharides. First of all, we have here glucose, alpha-D glucose, D because all are D stereoisomers. And here also we have alpha, this hydroxyl group. If someone asks about the hydroxyl group, it is always the hydroxyl group carried by carbon number 1. Carbon number 1 is the carbon that carries the functional group. You studied in the previous year how to number the carbon atoms. We number the carbon atoms so that the carbon atom having or linked to a functional group such as aldehyde group here, it must have the lowest number. Okay, back to disaccharides. So here it is an alpha-D glucose, because of this hydroxyl group that is linked to carbon number 1. When it makes a bond, there is elimination of a water molecule, and the bond that is formed here is named glycosidic bond. It is a covalent bond, of course. It is a glycosidic bond. We must understand the meaning of alpha-1,4. Without memorizing it, we must only understand what is the meaning of alpha-1,4 glycosidic bond. Alpha means that this is an alpha isomer and this is an alpha isomer. Notice the hydroxyl group here and the hydroxyl group here. 1,4, carbon number 1 reacting with carbon number 4. Carbon number 1 reacting with carbon number 4. This is maltose actually. This is a disaccharide named maltose. You must know that maltose is made up of 2 alpha-D glucose. Lactose is another disaccharide that is present in milk. It is made up of 2 hexoses, 2 pyranoses, but one is galactose, the other is glucose. Okay? The hydroxyl group is beta, position beta. This is the beta isomer, and this is also a beta isomer. Without explaining, you are not supposed to recognize the details now, just the general concept. In this other case, saccharose is a dimer. It is glucose alpha with fructose, glucose with fructose. So these are 3 types of disaccharides. You must know them. You must, without memorizing the chemical structures, you must be able to tell us which type of monosaccharides occur in each of these 3 types of disaccharides. Let's move now to polysaccharides. We don't consider examples of trisaccharides. We don't consider examples regarding oligosaccharides, other than this one actually. The blood groups may be considered as an oligosaccharide. Anyway, we don't consider neither trisaccharides nor oligosaccharides now. we consider the polysaccharides that may be homopolysaccharides or heteropolysaccharides. The homopolysaccharides are polysaccharides that are made up of a single type of monosaccharide. We have starch, also named amylopectin, glycogen, and cellulose. I just told you that cellulose is made up of beta-D-glucose, while starch and glycogen are made up of alpha-D-glucose. Cellulose is not branched. It forms polymers. That are not branched. Cellulose is a polymer of beta-D-glucose that is not branched. The bonds, the glycosidic bonds are 1,4 between carbon 1, carbon 4, carbon 1, carbon 4, carbon 1, carbon 4. In case of a glycogen and starch, we have alpha 1,4, 1,4, 1,4 alpha glucose, and we have a branching. At the branches, it is not 1,4. At the branches, it is 1,6. Carbon number 1 with carbon number 6. I repeat, you are not supposed to memorize the position of the glycosidic bond. You are just asked to understand the meaning of alpha, 1, 6, etc. Clearly, the branching pattern is different between glycogen and the starch. There are primary branches, secondary branches, while for starch we have only primary branches. These are examples of homopolysaccharides. Two figures that illustrate this is an electron microscopy image in the liver. It shows the glycogen granules in a liver cell, glycogen granules. Here an electron microscopy image of a chloroplast that contains starch granules. Chloroplasts sometimes show a certain number of starch granules. Okay? And cellulose forms fibers, as I said, not the granules. It forms fibers because it is long, unbranched, unlike glycogen and starch, which are branched. You expected after homopolysaccharides, heteropolysaccharides. I know that. But before I explain heteropolysaccharides, we need to learn about the possible modification of sugars. This is important before we undertake explanation of the heteropolysaccharide. This will be explained in the next video, inshallah.